Hello, so we have a biology paper here, which is of November 2024, paper 4.3. Okay, so we will navigate through this paper and see what it actually wanted. Okay, so let's start with question number 1. Number 1A says figure 1.1 shows part of the human digestive system. So we can see here, this is the diagram that is showing parts of the human digestive system. All right, what does the question want? Okay, so the question says, using label lines and letters shown, identify on figure 1.1 the organ that produces bile and label it as A, and the organ that stores bile and label it as B. Okay, so we know that the organ that produces bile is the liver, and the organ that stores bile is the gallbladder. So from there, we have to identify liver and we have to identify gallbladder from the diagram. Alright, so let's point out these things. Alright, so the liver is this one here. We are going to label it as A because it's the organ that produces bile. And the gallbladder, which stores bile, we are going to label it as B. So it will be labeled as such. Alright, yes, let's move to the next part. Describe physical digestion. So the next question wants us to tell what is physical digestion. So physical digestion is the process in which food substances are broken down into smaller substances without any chemical change. Okay, so this will say is the breakdown of food substances into smaller substances without chemical change. Alright, so food is being broken down but without any chemical change. Yeah, so this is what can be defined as physical digestion. Then the next question says, state the two places where physical digestion occurs in the body. So we have two places where chemical digestion is taking place. First of all, it takes place in the mouth by the action of teeth, but also it takes place in the stomach or in the small intestine by the action of what? Of bile. So we have the mouth, we're going to name the mouth over here, but we also have stomach. So these are places where physical digestion takes place. Alright, so let's see the next question. Bile is involved in fat digestion. Complete the sentences about fat digestion. Bile is released into the dash in the digestive system. The bile increases the surface area of fats and oil. This is dash. This increase in surface area increases the rate of that digestion using the enzyme which we have to state. Okay, so bile is usually released into the duodenum. Okay, it's usually released into the duodenum in the digestive system. Bile increases surface area of fats and oil. This is called emulsification. It's called emulsification. This increases surface area, increase in rate of chemical digestion. Alright, it increases the rate of chemical digestion using an enzyme known as lipase, because lipase is the enzyme that acts on what? On fats. Okay, then the next question says, state the name of the products of fat digestion. So when fats are broken down, what do we actually get? Alright, so when they are broken down, we get fat acids and glycine. Fat acids and glycine. So these are the products of fat digestion. Explain the role of bile in breakdown of protein by trypsin. Alright, so breakdown of protein by trypsin takes place in the duodenum of the digestive system. Alright, bile is not an enzyme, so it is not going to break down the proteins, but instead it is alkaline in nature. Alright, so if it's alkaline in nature, it is going to neutralize the acidity of the mixture that is coming from what? From the stomach. Alright, yes. So we'll say that bile neutralizes the mixture from the stomach. type skin activity. This is what it does. It neutralizes the mixture from the stomach so as to enable trypsin to work. Aphids are arthropods. Aphids insert their mouthpiece into phloem in the stem of the plant to feed. Alright, that is what they do. These are aphids. Okay, so let's see. As we can see there, we have the diagram of the aphid. Alright, yes. Identify two features visible in 2.1 that can be used to classify aphid as an insect. Now, what are the features that helps us to classify an aphid as an insect? Now, as we can see here, this diagram shows that the insect is having three body parts. It has this part here, it has this part here, and it has this part over here. Alright, but also we can see the legs, number of legs here, here, there. Alright, yes. So we see that it has three body parts, but also it has three pairs of legs. Those are features that can be used to classify it as an insect. So it has three body parts. Alright, but then again it has three pairs of legs. Yes, those are some features. Okay, then we go further. Fig 2.2 is a diagram of a cross section of the dicotyledonous stem. Okay, so we can see the dicotyledonous stem has parts labeled J, L, M, N. And then the first question says, state the letter in Fig 2.2 that identifies the structures from which aphids feed. So definitely they are feeding from the phloem, alright? And the phloem is part M, so we are going to label this M here. State the names of two nutrients transported in the phloem. Alright, so we need to know what is usually transported in the phloem, right? So what is transported in the phloem is like sucrose and amino acids. So we'll name that amino acids. Alright, so we go further. Scientists investigated the effect of temperature on the rate of translocation. The data are shown in table 2.1. Okay, right. Calculate the percentage change in rate of translocation between 15 degrees centigrade and 30 degrees centigrade. Give your answer to three significant figures. Alright, so we need to see what was there at 15 degrees and what was there at 30 degrees. Okay, alright, so at 30 degrees it was 17.1, and then at 15 degrees it was 7.3, but we have to check for the percentage change, right? Okay, so we are going to read the rate of translocation at 30, which is 17, and then we also take the rate of translocation at 15, which is 7.3. So it's going to be 17.1 minus at 15 to 7.3 over 7.3. The whole answer is going to be multiplied times 100, so we get it in percentage, so that we can obtain it in percentage. Okay, yes. Alright, so let's do it. When you work it out, you're going to get almost 134%. Alright, so this can be obtained as well as the percentage from that workout. Alright, then we move further. Fig 2.3 shows a potato plant growing in the spring and then in the summer. Alright, so we can see it as it is growing in the spring and as it is growing in what? In the summer. Alright, yes. So in spring, as you can see, it's like a potato tube, but then you have a growing shoot. Okay, and then in summer, you can definitely see shoots, stuff like that. Alright, yes. So let's move. It says here that explain how translocation changes between spring and summer in potato plant shown in figure 2.3. Alright, how translocation changes between spring and summer during this period of what? Of time. Use the time source and sink. Alright, so if you go back to the diagram here, we can see during spring, the growing shoot is coming from what? From the potato tuber. So it is actually that tuber is acting as a source here. It is acting as a source, providing a substance needed for growth by the what? By the shoot. Alright, yes. So we can say here, in spring, the potato tuber potato tuber is a source. Is a source to the growing shoot. Alright, providing it with substances needed for growth. But then again, we can look at what is happening in summer. Alright, in summer it means the leaves or the shoots here are the sources. They're involved in the process of photosynthesis to provide substances for the what? For the tuber. Alright, yeah. So we say that in summer, leaves are the sources. Okay, which means they photosynthesize. Photosynthesis occurs in leaves, right? And potato tubers are sinks, which means now they store what is being produced in the whole process of photosynthesis. Unlike spring, when it was spring, the potato tuber was a source while the shoot was a sink. And in summer, the potato tuber is now a sink while the shoots or the leaves are sources. Okay, new tubers form on the potato plant in summer. These tubers are genetically identical. Identify the type of reproduction that the potato plant is using to form tubers. Now, if they are genetically identical, it means the type of reproduction involved here is a sexual reproduction. That's what we are going to feel over here. Yes. So it will be a sexual reproduction. Alright. Yes, then we move to the next question. HIV can be transmitted from mother to infant. Describe how the risks of mother transmitting HIV to infant can be reduced. Alright, now how can we reduce the rate of transmission of HIV from a mother to a child? Alright, yes. So one of it is by testing pregnant women with HIV. Alright, so if we test a woman with HIV and maybe find her positive, we'll know how she will be advised to care for the pregnancy. Alright, yes, but also they'll be guided on, the doctors will know how to guide and deliver the child, but also C-section can be used instead of normal delivery. Alright, yes, it can be used, and also you can monitor the processes of breastfeeding. Maybe the mother will not be allowed to breastfeed their child. Instead, the child will only use bottled milk instead of using breastfeeding. Alright, yeah. So those are methods that can be used. Okay, scientists investigated the effect of breastfeeding on the chances of infants getting an infection. They studied two groups of 100 infants. One group was breastfed and one group was not breastfed. Type of infections, we can see here are the types of infections and number of individuals who are infected from those who are breastfed and those who are not breastfed. So you can see here. Alright, gastroenteritis, it had 4 for infants who are breastfed, and number of infants that were not breastfed, it was 8. Influenza, 5 for those who are breastfed and 4 for those who are not breastfed. Bronchitis, 7 for those who are breastfed and 9 for those who are not breastfed. Conjunctivitis, it is 12 for those who are breastfed and 10 for those who are not breastfed. Now, using the data in table 3.1, describe the effect of breastfeeding on the chance of getting an infection. Alright, so breastfeeding reduces the chances of getting bronchitis. We can say here, breastfeeding reduces the chances of getting bronchitis and gastroenteritis. This is the effect of what? Of breastfeeding. Alright, while we can see that with influenza and conjunctivitis, the children who are not breastfed were the ones who were having high risk. Alright, so it reduces the chances of getting these diseases. While bronchitis is infecting children or infants who are not breastfed. Alright, so this is the issue. Those who are breastfed are getting less chances of being infected, while the ones who are not breastfed are getting high chances of being infected. Alright, let's move further. Say the name of the blood cell involved in human respiration is usually known as white blood cell. So we'll write here white blood cells. Then describe how infants can gain passive immunity. So passive immunity, this is the type of immunity that we get from other individuals. Alright, so we'll say first, passive immunity is obtained from other individuals. Alright, then an infant obtains it from breastfeeding from their mothers. Alright, so this is one way in which they get what we call passive immunity. But also another way in which they get passive immunity is antibody injection. Alright, so these antibodies are obtained from elsewhere and then they are being injected into this person. Okay, describe how immunity gained by vaccination differs from passive immunity. So immunity from vaccination is an active immunity. Alright, it is an active immunity. Alright, it is not a passive immunity anymore. It is an active immunity. And why would we say that? It is because vaccines contain. This is because vaccines contain dead pathogens causing a disease. Alright, then these pathogens activate the immunity to produce specific antibodies. So you can see it is a form of immunity that is resulting first of all from pathogens entering the body and then the body making specific antibodies to it. That is active immunity. Well, it doesn't happen so in passive immunity. But also it forms or it makes memory cells. Alright, so this also makes it different from what? From passive immunity. Alright. Then we move further. Human muscle cells use anaerobic respiration during vigorous exercises. State the word equation for anaerobic respiration in humans. So anaerobic respiration in humans usually produces lactic acid. Alright, so if we are to state the equation, it will be that glucose is broken down to lactic acid. Yeah. Compare the process of anaerobic respiration to aerobic respiration in human muscle cells. Alright, so anaerobic to aerobic. So we say that anaerobic occurs in absence of oxygen. Anaerobic occurs in absence of oxygen, while aerobic occurs in presence of oxygen. That's one thing. Another thing is that in muscle cells, anaerobic forms lactic acid as a product. Alright, lactic acid. But aerobic forms carbon dioxide, and water. That's another difference. Another one is that anaerobic yields less energy than aerobic. Alright, so those are some comparison between aerobic and anaerobic. And then an athlete runs a race which lasts 60 seconds. The 4.1 shows the heart rate and breathing rate of the athlete during the race as he recovers from the race. Alright, yes. Then we see here the graph of the athlete as they are running. Alright, so you can see the heart rate, you can see the breathing rate. Okay, so the heart rate, for example, it was at almost 60, almost 62 or 60. Right, but then again it increased to almost 186 here after some seconds, after almost 50 seconds, while also the breathing rate was at 120, but it also rise to a certain point. Right, then after that we have a recovery period. Then after that you see a fall in heart rate and breathing rate. So let's see what the question wants. Okay, says that the rate of breathing increases during the rest in response to a change detected by the body. Identify the stimulus in this response and state the organ that detects the stimulus. Alright, so the stimulus here is increase in concentration of carbon dioxide. Alright, so as carbon dioxide concentration increases, it means there will be an increase in breathing rate, so more oxygen gets into the body. So the stimulus will be carbon dioxide concentration. Carbon dioxide concentration will stand as the stimulus, and the organ that is going to detect the change is the brain. Alright, using the information, calculate the rate of decrease in heart rate from 125 seconds to 260 seconds of an athlete. So heart rate at 125 and heart rate at, let's say, at 125 heart rate and at 260. Let's read from the graph. From the graph, we find that at 125 over here, the reading was, which is like 106. Alright, so we'll go back here and say at 125 it was almost 180, 182, and then here it was 106. So using the information, calculate the rate of decrease in heart rate from 125 seconds as the athlete recovers. Okay, so when we work out this one in our calculator here, we find it to be almost 0.53. Alright, yes, so that is the heart rate per minute. Okay, then we move further. Explain the results shown in fig 4.1 from 60 seconds until 420 seconds. Alright, so we have to explain what is shown in that diagram from 60 to 420. Where's the diagram again? So 60 is almost here, where the heart rate is like at 186, and then to 420. 420 is like when the heart has returned to its normal rate. 60 seconds, the heart rate is at the highest level, right? But at 420, the heart rate is at its what? Normal level. We'll write it. Okay, so at 60 seconds, the heart rate is at highest level. Okay, that's one thing. But apart from that, we know that at 420 seconds, the heart rate is at normal level. The heart rate is at normal level. It has returned to its normal level. That is what is expected only. Alright, yeah. But then again, we need to know why is the heart rate at such a level. Okay. And why doesn't the heart rate return to normal just after 60 seconds, just after the exercise have ended? All right, yeah. Okay, so again we'll say heart rate from the diagram does not return to normal level immediately. Alright, heart level does not return to normal level immediately, so that more oxygen, or so that oxygen is much allowed into the body, so that someone breathes in rapidly and gains in more oxygen. Alright, yes. Then again, at high levels, at high heart rates, it means there is high rate of aerobic and anaerobic respiration. Anaerobic respiration, which producing lactic acid. So at high rates, we expect formation of what? Of lactic acid. Alright, yes. But then again, after high rates, then when the rate of breathing returns to normal, heart rate returns to normal, lactic acid is what? Is taken to the liver and completely broken down to carbon dioxide and water. Alright, yes. We say that after 60 seconds, lactic acid is carried to liver, to be broken down, to be completely broken down. That's another thing that will take place during this period of time. And also, yes, I spoke about the breathing rate. The breathing rate is high at 60 seconds and returns to normal at 420. In this graph, from the period of 60 seconds to the period of 420 seconds. Alright, let's go further. Number 5 says feather color in some breeds of chicken is an example of codominance in birds. In this example, a chicken with white feathers and a chicken with black feathers can breed to produce offspring that have white feathers and black feathers. The allele for black feathers is F with a B, and the allele for white feathers is F with a W. 5.1 is the pedigree of a family of chicken. You can see that we have this female over here, which is white, and we don't have the allele for the male. Then we can see the offsprings, which are 4 and 5. They are all black and white. Okay, they have black and white color. Okay, then we have the key here. The black ones represent black feathers, the white ones represent white feathers, and the one with black dots in them represent chicken with both black feathers and white feathers. Alright, then the first question says, state the type of variation that is caused by genes only. Okay, so the type of variation that is caused by genes only is usually discontinuous. We will say discontinuous variation. It's a kind of variation that is caused by genes only. Then state all the possible genotypes of chicken 2 and chicken 7. Chicken 2 and chicken 7. Where's chicken 2? Alright, so chicken 2 is probably a black chicken. Why? Because the offspring, which are 4 and 5, have what? Have white and black feathers. So we'll say the genotype for chicken 2 is going to be black. Alright, which will be FB. Alright, then chicken 7. What is chicken 7? chicken 7. All right, so chicken 7 is like a breed of white and black. All right, so if it's a breed of white and black, it means if we do the pedigree diagram, it's going to be like this way. We have black, white, and here we have black, white. What will the options be? To be black with black, we will get black and white here. We'll get again black and white here, and then we'll get white. So the possible genotypes of chicken 7 can be black and white, or can be white. So that's what we are going to write over here. Can be black and white, but it can also be white. Yes, those are the possible genotypes. All right, then we go to the next question. Complete the genetic diagram to show the results of a cross between chicken number 5 and chicken number 6. We need to see chicken 5 phenotypes and genotypes. Okay, chicken 5 is here, it's black and white, and chicken 6 is totally black. All right, yes. So chicken 5 phenotypes, it's a white, or chicken 6 was black, phenotypical, and then for the chicken 5 it also black and white. So right here, black and white, that's how it appeared phenotypical. Now if we are to write the genotypes, it means this will be black and white, while this one here will be black. Alright, so the gametes will be black, white, while the gametes here will be black. Alright, so complete the genetic diagram to show a cross between these two. Okay, so you have to show offspring genotype expected. So when we do a pedigree diagram, for example, over here, what do we expect to get? We had a black and white, and here we had a black chicken. So this one will give us black first. This one here will also give us black. This one here will give us black and white. And that one will give us black and white. Alright, so offspring's genotype will be either black or black and white. Expected phenotype, which will be black or black and white. Alright, state one example of codominance in humans. Okay, one example of codominance that we can see in humans is ABO blood groups. Example, blood group AB. It's a very good example. Alright, then we move to the next question. Some blood clotting disorders can be inherited. Describe what is meant by the term inheritance. So inheritance is the transmission of genetic information from one generation to another. We'll define it as the transmission of genetic information from one generation to another. And then describe the process of blood clotting. So the process of blood clotting, first of all, occurs due to the presence of red blood platelets. Alright, then what do the red platelets do? They stimulate the production of fibrinogen, which is soluble. Fibrinogen is a kind of protein. Then fibrinogen is converted into fibrin, which is insoluble. Then fibrin forms mesh-like structures that trap blood and allow it to clot. So blood platelets bring about allow clotting. How so? Through production of fibrinogen, which is soluble. Then again, fibrinogen is changed into fibrin, and you know that fibrin is insoluble. Alright, then fibrin forms mesh that trap blood allowing blood clot to occur. Alright, then we have here B. Genetic modification has been used to produce human proteins. One protein made in this way is factor 7A, which is used to treat some blood clotting disorders. Fig 6.1 shows parts of the process. You have human cells, you have DNA extracted, and DNA cut with enzyme A. The other side, you have plasmid cut with enzyme A again. Then cut DNA and cut plasmid are joined together using enzyme B to form C. Then C is inserted into a bacteria. After it's inserted into the bacteria are gone in a fermenter to produce factor 7A. Alright, state the name of enzyme A and enzyme B. So enzyme A is used to cut. Yes, and then enzyme B is used to join them together. So enzyme A is restriction enzyme. Restriction enzyme. It is the enzyme that is used to cut. And then B is DNA ligase. It's an enzyme that is used to seal